Hello and welcome to the massive open online course on human genetics. Today we are going to discuss an important topic about gene expression. The main objectives of this module are to know the concept of gene expression, to know the basic mechanism of gene transcription, to know the process of protein translation. All cells control when and how much each one of its genes are expressed. First thing, we need to understand what it means when we say that a gene is expressed. If the gene encodes a protein, one might reasonably propose that expression of a gene means how much functional protein is made. But what if the gene does not encode a protein but rather some functional RNA? Then in this case, expressed or expression might mean how much of the functional RNA is made. Genes encode proteins and proteins dictate cells' function. Therefore, the thousands of genes expressed in a particular cell determine what that cell can do. Therefore, we can define that gene expression is the process by which information from a gene is used in the synthesis of a functional gene product that enables it to produce end products like proteins or non-coding RNA and ultimately affect a phenotype as the final effect. These products are often proteins, but in non-protein coding genes such as transfer RNA, that is tRNA, and small nuclear RNA, that is snRNA, the product is a functional non-coding RNA. In a nutshell, gene expression is the process of transcription of a gene into mRNA, the processing of that mRNA and its translation into the protein for protein encoding genes. And gene expression is summarized in the central dogma of molecular biology, first formulated by Francis Crick in 1958, further developed in his 1970 article and expanded by the subsequent discoveries of reverse transcription and RNA replication. The central dogma of biology is that DNA is used as a template to transcribe RNA, and RNA is used to synthesize proteins, and those proteins give you your unique phenotype of physical characteristics. And different cells turn on and turn off different DNA sequences by a process called the differentiation. Gene expression consists of multiple steps that are dynamically and closely regulated. Gene expression is a tightly regulated process that allows a cell to respond to its changing environments. Regulated expression of gene is required for adaptation, differentiation, and development. Epigenetics is one of the ways how gene expression is regulated. RNA also plays a very important role in gene regulation, as the gene's activity is controlled first and foremost at the level of transcription. In addition to transcription level controls, gene expression can be modulated by gene amplification, gene rearrangement, post-translational modifications, and RNA stabilization. In genetics, gene expression is the most fundamental level at which the genotype gives rise to the phenotype, that is the observable trait. The genetic information stored in DNA represents the genotype, whereas the phenotype results from the interpretation of that information. Such phenotypes are often displayed by the synthesis of proteins that control the organism's structure and development, or that acts as enzyme catalyzing specific metabolic pathways. There are two key steps involved in making a protein. One is transcription and the second is translation. Dear students, let's now discuss the transcription. The initial step in gene expression is the transcription of the DNA molecule into an exact RNA copy. The transfer of information to the ultimate synthesis of a protein is accomplished via an RNA intermediate, the so-called messenger RNA. The mRNA molecule contains the exact same sequence of nucleotides as found in the DNA molecule, with U, that is uracil, substituted for thymine. This occurs through the process known as transcription and is carried out by an enzyme termed as DNA-dependent RNA polymerase. The product of transcription is an RNA molecule that is identical in sequence content to one of the DNA strands, the sense strand, and the complementary to the other DNA strand, the template strand. The RNA differs from DNA in two respects. First is RNA contains a hydroxyl group residue at the 2' position of the sugar moiety, that is the ribose sugar, whereas DNA contains a hydrogen atom at this position, that is the deoxyribose sugar. This changes some of the chemical properties of the two molecules. Second RNA replaces the base thymine with uracil. Although they are chemically different, their base pairing properties, that is complementarity to adenosine, are the same. Thus, in DNA, adenosine pairs with thymine, whereas in RNA, adenine pairs with uracil. Transcription always proceeds in a 5' to 3' direction with respect to polarity of the nucleotides in the RNA. Thus, an unmodified primary transcription product would contain a 5' end with a triphosphate and a 3' end with hydroxyl atom or OH group. There are actually three distinct forms of RNA polymerase found in eukaryotic cells that are responsible for the transcription of three distinct types of genes. The genes encoding the ribosomal RNAs are transcribed by RNA polymerase 1. The protein encoding genes that produce the messenger RNAs are transcribed by RNA polymerase 2. Finally, the genes encoding transfer RNAs as well as certain other small RNA molecules are transcribed by RNA polymerase 3. Transcription involves three basic steps: the initiation, elongation, and termination. Let's first discuss the initiation. RNA polymerase binds to a sequence of DNA called the promoter region, found near the beginning of a gene. Each gene or group of co-transcribed genes in bacteria has its own promoter. Once bound, RNA polymerase separates the DNA strand, providing the single-stranded template needed for transcription. Now the elongation step. One strand of DNA, the template strand, acts as a template for RNA polymerase to move. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotides, making a chain that grows from 5' to 3'. The RNA transcript carries the same information as the non-template, that is coding strand of DNA, but it contains the base uracil instead of thymine. That is, thymine is replaced by uracil in the RNA molecule. Now let us discuss the termination step. Sequences called terminators signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase enzyme. The three steps involved are accomplished via the interaction of a variety of proteins called transcription factors that provide the recognition step, guiding the polymerase to the correct site. Moreover, the interaction of these transcription factors with their DNA recognition sequences represents a rate-limiting step in the process of transcription initiation. Sequences in the DNA which are recognized by these transcription factors and which serve as a binding sites for the transcription factors are generally upstream, that is 5' of the start site for transcription, and are called as promoters. Additional transcription factors can bind to sequence elements called enhancers that may be located further upstream or even downstream of the gene. In either case, the DNA sequences that bind the transcription factors must be located on the same DNA molecule, thus the same chromosome as the gene which is regulated. As such, these sequences are said to act in cis conformation. The promoter is absolutely essential for transcription, whereas the enhancer, as its name implies, increases the efficiency of transcription. A critical component of many promoters is the sequence element, that is the Tata box, which is located nearest to the site of transcription initiation, about 30 nucleotides away. The Tata box element binds a general transcription factor known as TBP, that is Tata binding protein, which is a component of a larger complex known as TFIID. Now let's discuss the steps involved in transcription initiation. Although variations in any step of gene expression can be regulatory, by far the most frequent form of gene control is the regulation of transcription initiation. Control of transcription involves the regulation of transcription factors that interact with the critical cis-acting sequences in the promoter and enhancer that then dictate the frequency of RNA polymerase binding to that gene and subsequent transcription process. The process of promoter recognition and utilization involves a stepwise interaction of a complex series of transcription factors with the promoter to create a stable DNA protein complex that allows RNA polymerase to initiate transcription. Key in this process is the recognition of the specific sequences in the promoter DNA sequence, the cis-acting elements, by the trans-acting transcription factors. Given the fact that there are as many as 100,000 protein encoding genes in the mammalian genome, it is obvious that the expression of each gene cannot be regulated by unique transcription factors. Rather, it would appear that a limited number of transcription factors are responsible and that the high degree of specificity is generated by specific protein-protein interactions that stabilize otherwise weak interactions on a promoter. Very likely, the ability of specific factors to interact provides the possibility of combinatorial interactions of promoter-specific factors that create specificity to transcription control. Although it was initially believed that the three types of eukaryotic genes, that is polymerase I or POL1, ribosomal RNA, POL2 mRNA, or POL3, that is tRNA, were quite distinct with respect to factors involved in the transcription initiation event. It is now clear that many of the activities involved in the promoter recognition are shared. Moreover, although there are distinct RNA polymerases for the three types of genes, many of the subunits of the polymerases are also shared. What distinguishes the classes of genes most clearly is the complexity of regulatory elements and factors necessary for the transcription of the mRNA genes. As stated before, transcription factors possess two essential properties: the ability to bind to DNA recognizing the cis-acting elements in a sequence-specific manner and the ability to stimulate transcription. The fact that these two properties are a function of distinct and separable domains in the protein has been exploited in a method to detect protein-protein interactions, the so-called yeast 2 hybrid assay, in which the DNA sequences encoding the two functional domains of a yeast transcription factor have been separated in two vectors. Sequences are then cloned into these vectors to create chimeric fusion proteins as a method for selecting or assaying for sequences that will bring the two functional domains back together via protein-protein interactions. Dear students, let's now discuss the post-transcriptional events of gene expression. In prokaryotes, only one type of RNA polymerase catalyzes the transcription of all types of RNA as compared to that of the eukaryotes where three different RNA polymerases were involved. Prokaryotic mRNA does not require to be processed to be functional and synthesize a protein. Transcription and translation both occur in the cytosol and can occur simultaneously in bacteria. So translation can even start before the mRNA is fully transcribed. In eukaryotes, the primary RNA transcript undergoes structural and chemical change after transcription to become a mature and functional RNA, which is then transported out of the nucleus to perform various functions. These modifications are called post-transcriptional modifications. RNA polymerase second produce hnRNA or heterogeneous nuclear RNA, which is the precursor of mRNA molecule. hnRNA is non-functional and undergoes post-transcriptional modifications to produce the mature mRNA. The three main post-transcriptional modifications are splicing, capping, and tailing. Now let us first discuss the splicing. In eukaryotes, the structural gene has coding and non-coding regions. Introns are removed and coding regions or exons join to form a mature mRNA. Now let us discuss the capping process. At the 5' end of the heterogeneous nuclear RNA is methyl guanosine triphosphate. It provides the cap to the mRNA molecule. Now the tailing. At the 3' end on the tail side end of the heterogeneous nuclear RNA is a polyadenylation sequence, that is many adenine units are attached. This forms the polyadenylation tail and is about 200 to 300 bases long. The mature mRNA produced after the processing is transported to the cytoplasm for protein synthesis. Post-transcriptional modifications are an important point at which gene expression is regulated. Dear students, this was all about transcription. Now let us discuss the another important process, that is the translation. In translation, messenger RNA or mRNA is decoded in a ribosome outside the nucleus to produce a specific amino acid chain or polypeptide. The polypeptide later folds into an active protein and performs its function in the cell. The ribosome facilitates decoding by inducing the binding of complementary tRNA, that is anticodon sequence, to mRNA codons. The tRNA, that is transfer RNAs, carry a specific amino acid that are chained together into a polypeptide as the mRNA passes through and is read by the ribosome. Translation proceeds in three phases: the initiation, elongation, and termination. Let us first discuss the initiation. The ribosome assembles around the target mRNA first. Then the first tRNA is attached at the start codon. Then the elongation. The last tRNA, validated by the small ribosomal subunit, that is accommodation, transfers the amino acid it carries to the large ribosomal subunit, which binds it to the one of preceding admitted tRNAs, that is transpeptidation. The ribosome then moves to the next mRNA codon to continue the process of translocation, creating an amino acid chain. Then the third step, that is the termination. When a stop codon is reached on the mRNA, the ribosome releases the polypeptide chain. The ribosomal complex remains intact and moves on to the next mRNA to be translated. In prokaryotes, bacteria and archaea, translation occurs in the cytosol, where the large and small subunits of the ribosome bind to the mRNA. In eukaryotes, translation occurs in the cytoplasm or across the membrane of the endoplasmic reticulum in a process called co-translational translocation. In co-translational translocation, the entire ribosome mRNA complex binds to the outer membrane of the rough endoplasmic reticulum, and the new protein is synthesized and released in the endoplasmic reticulum. The newly created polypeptide chain can be stored inside the endoplasmic reticulum for future vesicle transport and secretion outside the cell, or immediately secreted. Many types of transcribed RNAs such as transfer RNA, ribosomal RNA, and small nuclear RNA do not undergo translation into proteins. Dear students, let's now discuss the basic mechanism of translation. The basic process of protein production is addition of one amino acid at a time to the end of a protein. This operation is performed by a ribosome. A ribosome is made up of two subunits, a small subunit and a larger subunit. These subunits come together before translation of mRNA into a protein to provide a location for translation to be carried out and a polypeptide to be produced. The choice of amino acid type to add is determined by an mRNA molecule. Each amino acid added is matched to a 3-nucleotide subsequence of the mRNA. For each such triplet possible, the corresponding amino acid is accepted. The successive amino acids added to the chain are matched to successive nucleotide triplets in the mRNA chain. In this way, the sequence of nucleotides in the template mRNA chain determines the sequence of amino acid in the generated amino acid chain or the polypeptide. Addition of an amino acid occurs at the C terminus of the peptide, and thus translation is said to be amine to carboxyl directed, that is from amine group to carboxyl group. The mRNA carries genetic information encoded as a ribonucleotide sequence from the chromosome to the ribosome. The ribonucleotides are read by translational machinery in a sequence of nucleotide triplets called the codons. Each of those triplet codes for a specific amino acid. The ribosome molecule translates this code to a specific sequence of amino acids. The ribosome is a multi-subunit structure containing rRNA and proteins. It is the factory where amino acids are assembled together into proteins. tRNA are small non-coding RNA chains, that is usually of the size of 74 to 93 nucleotides, that transport amino acids to the ribosome site. tRNA have a site for amino acid attachment and a site called as anticodon. The anticodon is an RNA triplet complementary to the mRNA triplet that codes for their cargo amino acid. Amino acyl tRNA synthetase, that is an enzyme, catalyzes the bonding between the specific tRNA and the amino acid that their anticodon sequences call for. The product of this reaction is a amino acyl tRNA molecule. In bacteria, this amino acyl tRNA is carried to the ribosome by an elongation factor EFTU, where mRNA codons are matched through complementary base pairing to specific tRNA anticodons. Amino acyl tRNA synthetases that mispair tRNA with the wrong amino acids can produce mischarged amino acyl tRNAs, which can result in inappropriate amino acids at the respective position in protein. This mistranslation of the genetic code naturally occurs at low levels in most organisms, but certain cellular environments cause an increase in permissive mRNA decoding, sometimes to the benefit of the cell. The ribosome has two binding sites for tRNA. There are the amino acyl site abbreviated as A, the peptidyl site or exit site abbreviated as P/E. With respect to the mRNA, the three sites are oriented 5' to 3' E, P, and A. Because ribosomes move towards the 3' end of the mRNA, the A site binds the incoming tRNA with the complementary codon on the mRNA. The PE site holds the tRNA with the growing polypeptide chain. When an amino acyl tRNA initially binds to its corresponding codon on the mRNA, it is in the A site. Then a peptide bond is formed between the amino acid of the tRNA in the A site and the amino acid of the charged tRNA in the P/E site. The growing polypeptide chain is transferred to the tRNA in the A site. So translocation occurs moving the tRNA in the PE site now without an amino acid. The tRNA that was in the A site, now charged with the polypeptide chain, is moved to the PE site, and the tRNA leaves and another amino acyl tRNA enters into the A site to repeat the process again and again so that a long polypeptide chain is formed. After the new amino acid is added to the chain and after the tRNA is released out of the ribosome and into the cytosol, the energy provided by the hydrolysis of GTP bound to the translocase EF, that is elongation factor G in case of the bacteria, and elongation factor 2 AE in eukaryotes and archaea, moves the ribosome down one codon towards the 3' end. The energy required for translation of proteins is significant. For a protein containing n amino acids, the number of high energy phosphate bonds required to translate is 4n-1. The rate of translation varies. It is significantly higher in prokaryotic cell. Up to 17 to 21 amino acid residues per second are added. Then in eukaryotic cells, where up to 6 to 9 amino acid residues per second are added to the growing polypeptide chain. Even though the ribosomes are usually considered accurate and processive machines, the translation process is subject to errors that can lead either to the synthesis of erroneous proteins or to the premature abandonment of translation, either because a tRNA couples to a wrong codon or because a tRNA is coupled to a wrong amino acid. The rate of error in synthesizing proteins has been estimated to be in between 1 in 10 raised power 5 and 1 in 10 raised power 3 misincorporated amino acids depending on the experimental conditions. The rate of premature translation abandonment instead has been estimated to be of the order of magnitude of 10 to 4 events per translated codon. The correct amino acid is covalently bonded to the correct transfer RNA or tRNA by amino acyl transferase enzymes. The amino acid is joined by its carboxyl group to the 3' or OH hydroxyl group of tRNA by an ester bond. When the tRNA has an amino acid linked to it, the tRNA is termed charged. Initiation involves the small subunit of the ribosome binding to the 5' end of mRNA with the help of initiation factors. In bacteria and a minority of archaea, initiation of protein synthesis involves the recognition of a purine-rich initiation sequence on the mRNA called the Shine-Dalgarno sequence. The Shine-Dalgarno sequence binds to a complementary pyrimidine-rich sequence on the 3' end of the 16S RNA, part of the 13S ribosomal subunit. The binding of these complementary sequences ensures that the 30S ribosomal subunit is bound to mRNA and is aligned in such a way that the initiation codon is placed in the 30S portion of the P site. Once the mRNA and 30S subunit are properly bound, an initiation factor brings the initiator tRNA amino acid complex F-methionine tRNA, that is formyl methionine tRNA, to the 30SP site. The initiation phase is completed once a 50S subunit joins the 30S subunit, forming an active 70S ribosome that is larger in size. The termination of the polypeptide occurs when the A site of the ribosome is occupied by a stop codon UAA, UAG, or UGA, also known as EMBER, OCHER, or OPEL, respectively, on the mRNA, creating the primary structure of a protein. tRNA usually cannot recognize or bind to stop codons. Instead, the stop codon induces the binding of a release factor protein RF1 and RF2 that prompts the disassembly of the entire ribosome mRNA complex by the hydrolysis of a polypeptide chain from the peptidyl transferase center of the ribosome. Drugs or special sequence motifs on the mRNA can change the ribosomal structure so that near cognate tRNA are bound to the stop codon instead of the release factors. In such cases of translation read-through, translation continues until the ribosome encounters the next stop codon. The process of translation is highly regulated in both eukaryotes and prokaryotic organisms. Regulation of translation can impact the global rate of protein synthesis, which is closely coupled to the metabolic and proliferative state of a cell. In addition, recent work has revealed that genetic differences and their subsequent expression as mRNA can also impact translation rate in a RNA-specific manner. Dear students, it was all about today's lecture. Hope you have understood it well. See you with a new topic next time. Take care. Bye-bye.